Em prosseguimento ao nosso simpósio, daremos início à próxima mesa redonda, cujo tema é Surto de Ebola, um estudo de caso. Convidamos como moderadora e coordenadora desta mesa a professora da Universidade Federal de Goiás, que trabalha em desenvolvimento em teste de diagnóstico, professora Samira Birer. Representando o time de pré-qualificação de diagnóstico da Organização Mundial de Saúde, doutora Anita Santos. O diretor associado do Laboratório de Ciência do Centro de Controle e Prevenção de Doenças, CDC, Ron Ballard. A senhora representante do Serviço de Laboratório Nacional de Saúde da África do Sul, Sarvashni Mudliar. A senhora diretora adjunta do Departamento de Vigilância das Doenças Transmissíveis da Secretaria de Vigilância em Saúde do Ministério da Saúde, Vanessa Tenório de Oliveira. O senhor Javan Sfandiari, da Kembaio. Nessa mesa será discutido qual o papel do diagnóstico em epidemias, formação de recursos, capacity building, inovação versus acesso. Com a palavra, doutora Samira Birer.
Bom dia a todos, Carlos, obrigado pelo honroso convite e de coordenar uma mesa tão quente e todo mundo está interessado. Então, eu vou pedir aos palestrantes que permaneçam dentro do horário de 20 minutos para cada um, embora não tenha sido regulamentado antes, mas aproximadamente isso está bem, que a gente consegue vencer o tempo e dar um pouquinho de espaço para alguma pergunta que surja. Então, eu vou dar a palavra à doutora Anita para iniciar a apresentação.
Okay, so I hope you're not sick and tired of listening to me. This is the last time, I promise. I'm going to talk to you a little bit about a new activity that we have implemented in the last 6 weeks, actually, in WHO and within the pre-qualification team. So as you can imagine, WHO is sort of very busy at the moment with all of the aspects related to the Ebola outbreak. We have drug trials, vaccine trials, and we have issues related to diagnosis and laboratory, as well as all the epidemiology. And there's just really, we have one Ebola expert in WHO, one person who's a technical expert, and the rest of us have had to draw on the skills that we have and to be able to sort of mobilise to help. So people wonder why WHO is taking a little bit of time. That one Ebola expert is travelling from country to country. So what we've done is that we're very well aware that there is a great need for testing in particularly the 3 most affected countries, but also in neighbouring countries and obviously also even in our own home countries. The US, Australia has needed testing as well. And so there's actually a variety of diagnostics that you could use. We've elected to narrow the scope of our emergency quality assessment mechanism because we want to be able to provide results very, very quickly about priority diagnostics, which we think can really help to A, stop transmission, and B, make sure that those people who actually do have Ebola are treated appropriately. Unfortunately, Ebola has very non-specific symptoms. It's febrile illness, vomiting, diarrhea, any number of other common diseases in West Africa could cause these symptoms, malaria, obviously, typhoid and the like. And so we've elected to look at 2 types of diagnostics. One would be, actually I'll still keep talking, one would be antigen detection rapid diagnostic test, and that's a sort of test that could be used in a community setting, so in a person's home or at a health centre, where a person with a symptom, so febrile illness or vomiting or diarrhoea, could be tested. We know that these sorts of tests are not as accurate as the molecular methods, but we're really trying to find a test that could be used out in the community to exclude Ebola, at least as a cause of their symptoms. And so that would mean if we could exclude those people from having Ebola, they wouldn't then need to go to the holding centres to then be triaged into the Ebola treatment centres and Ebola care centres. Unfortunately, when somebody is sick and they go into triage, they may not have Ebola, but there might be one other person there that does have Ebola. And unfortunately, these holding centres have really become transmission centres, and a lot of people are actually acquiring Ebola because they're being quarantined. So we sort of take this part of it very seriously. Obviously, it's very difficult for families to get sick people to the places where testing can take place. Taxi drivers, motorbike taxis are reluctant to take unwell people. So we're really trying to see if these products could be useful. There are actually a lot of RDTs out there. We've heard of a few popping up in Nigeria and places such as this. However, very little is known about the quality or the safety or the performance of these tests. There's perhaps a little bit more information about the molecular tests, and obviously the molecular tests require a laboratory, they require very skilled staff, they require a certain amount of specimen preparation, and it's normally a reverse transcriptase PCR. However, at the moment, they're still a very complex, low throughput test, and so at the moment they're being used to screen and to confirm with a throughput of maybe, in some countries, only 70 specimens per day. For instance, were being tested in Liberia a month ago. So 70 people with febrile illness. So you can see why we need to think of alternate solutions. So we decided to develop this mechanism. It's not pre-qualification, it's absolutely not that. It's very much an emergency stopgap, and it's quality assessment. Most of the IVDs that we were able to find had very limited clinical performance data. They had not been tested in this most recent outbreak. They may have been tested on a previous outbreak, but generally not even. So we really did have assay developers preparing a test in their R&D laboratory and had never actually seen a clinical specimen. So that was really why we figured we needed to really do something. Also considering the burden in those affected countries, we don't want hundreds of rapid test developers and molecular test developers going there to be performing clinical trials when there's more pressing needs. So we do follow a similar sort of approach. We actually put out an expression of interest on the 18th of September. It was open for 1 month, so it's now closed. However, we are able to accept by exception submissions. We reached out to everyone that we knew that had a test, but we didn't know ChemBio had a test, so we're reaching out now. We do a very abbreviated dossier review, which means we ask manufacturers to submit us any data they have, so any analytical data, any stability data, whatever they have done to in some way validate their test. We do a desktop audit of quality management systems, so we ask for their ISO certification and certain quality documents. A lot of the manufacturers, certainly for the molecular side, have research use only assays. They've never sold clinical diagnostics, and very few have ISO 13485. We are going to conduct a laboratory evaluation. This is obviously a very difficult task because those sort of laboratories that have an appropriate biosafety level and the appropriate experts are very, very busy. So we're still working on that. And we hope that we can make a decision within 3 months. And the final decision would be made by an internal WHO committee, so clinicians, epidemiologists, Pierre Formonte, who is our Ebola expert, and others who have been brought in to help us with the crisis. And just recently we had someone from the Gates Foundation, who's actually ex-FDA regulator, who has joined our department specifically to monitor how we're doing this piece of work. So obviously we went and looked at what other regulators were doing, and it's pretty much only the US FDA, and they have an emergency use approval mechanism, and they were able to enact it for Ebola. They have 5 products, I think, that have been approved. One is a Department of Defense product. There is 2 CDC RT-PCRs and the BioFire from bioMérieux. The complexity with an EUA is that the manufacturer must specify which labs it would be performed in. So the DoD assay has to be performed by DoD, and the CDC assay has to be performed by CDC. So it's a little bit limiting in the way it's used. They don't look at any aspect related to the quality management system. So we're looking at ways to sort of work with them and look at certainly aspects related to the analytical performance and the clinical data that they may have. And as you can see, this is all the steps we use with pre-qualification, and there's much less here on this side. The European Union, it would have to be CE marked as and we only know of one product that is CE marked, which is the Altona Diagnostics RT-PCR. So as of the 1st of November, we have 15 applications. We have 4 RDTs for a detection of Ebola antigen, and we have 11. And I know that since I've been away, we've actually got even more applications. Obviously, we're interested if it has a CE mark or it has FDA EUA, we can expedite certain parts of the assessment. As I mentioned, we have very few of the diagnostics have ever been applied clinically. They're all research use only assays. Therefore, there's absolutely no or very limited clinical data. We obviously decided we then needed to do a lab evaluation. We at first wanted to be able to do it in a BL4, and we were hoping to do it perhaps not in the field, to do it in one of our European WHO collaborating centres, because we didn't want to overburden our labs in Sierra Leone and Liberia and Guinea, but they just didn't have enough specimens. I mean, when something like this is happening, it's very hard for people to stop, collect a specimen, catalogue it, and send it to a repository. We also are faced with a lack of international biological reference preparation to calibrate particularly the RT-PCRs and the antigen assay, actually. It's very difficult to establish a reference assay, particularly when the RT-PCRs might be all looking at different targets. And obviously, how do we assign a reference value for the antigen assays? So you can't really use a nucleic acid detection assay to generate the sensitivity and specificity of antigen. And obviously the ethics around all of this, about conducting a lab evaluation at a time of such crisis. So we have developed a protocol for both the antigen RDTs and for NAT. We've identified 6 potential labs. And as I said, we're really concerned about biosafety, and that's something that we want to make sure we're protecting the workers conducting the work. We're going to limit the evaluation to a few analytical parameters, so limit of detection for both the antigen and for the NAT. And for the antigen RDTs, also prozone effect. So when Ebola really gets going, you have a lot of viral particles and a lot of antigen, and you may get prozone, and it may appear the test is negative. And obviously looking at diagnostic sensitivity and specificity, and we will publish these data. So if an IVD is found to be acceptable, it passes our laboratory evaluation, and the other aspects are found to be acceptable, we would list it as eligible for WHO procurement for a time-limited period. We're also working with countries to develop testing strategies, or testing algorithms, as some people might call them. And at the moment we foresee, if we have a good Ebola antigen RDT, we could employ that as the screening test and then just reserve the molecular test for confirmation. However, if we have a very good NAT test that's very cheap and works very well and is easy to use, we might consider that we just use that for the screen. We've been presented with a whole range of products. I didn't bring any photographs, but we range from large instruments, sort of, you know, with whatever light cycler or instrumentation you have, to very small, sort of in a box. So they might be used at different levels of the health service. And we're actually really wanting to find a product that we can take out of the lab and put into the Ebola care unit and the Ebola treatment units. And at the moment, we don't really want to have to go through the full pre-qualification with these products. This is just an emergency that we've set up. We will have post-market surveillance. We're really concerned about the capacity to make a lot of money out there with creating fear around Ebola. And one of the other objectives of this emergency QA assessment is to exclude products that don't work well and to make sure that people are not paying $5 to $10 for a rapid test that is very, very insensitive. So we're going to be using the same post-market surveillance approach as I described yesterday to make sure any complaints are dealt with quickly. And if you have any questions about the work we're undertaking, as I said, we hope to have the results in 3 months' time. I know that's not quick enough, but you can always contact myself or anyone on our generic email address. Thank you.
Deixa as perguntas para o final. Então, Dr. Ron, por favor, sua vez.
Oh, no, not that one. No. It's all right, it doesn't matter. We can leave it. It's fine. This is a document which actually I have in my hand. It's a nice thick document, and it would be real fun to go through this document for you today. I think it really doesn't matter. Yeah. As a result of me going second, I had a lot of sort of basic stuff on Ebola, but I don't think that's necessary for this discussion. I just want to tell you a little bit about what CDC is doing around Ebola at the moment. You see, we have a responsibility primarily to the domestic side of our work, and so consequently, the development of a real-time reverse transcriptase PCR for Ebola was very important, and the distribution of those tests across the states to the state public health laboratories was really the priority for CDC initially. The CDC RT-PCRs and the kits absolutely have to be used on 1 or 2 platforms only. And that's one of the problems associated with these real-time PCRs. You can't necessarily use a kit on different platforms and expect to get the same answers. So I'm not sure what the platform is that the Canadians use. I'm not sure the platforms that the South Africans use, or the Chinese, or in fact the Russians, or the Japanese. But clearly there's a need to look at these platforms and to find some common ground, so that you don't have to buy another very expensive piece of equipment in order to be able to perform these tests. So, for instance, our tests are performed on applied biosystems 7500s or 7900 HT real-time PCR systems, and we even have to use specific Invitrogen superscript 3 platinum one-step tests in order to be able to use a validated SOP. And the SOPs are actually included in this wonderful document, which you can actually access if you really want to by going to our website, which is, surprise, surprise, it's www.cdc.gov. Very complicated. Our efforts in West Africa, on the other hand, are largely either related to epidemiology, contact tracing, logistics, and lab. So we do not have docs in the field who have patient contact. And I think that's one of the real problems, that either the US Army goes out there and they're logistics people, or they're doing a little bit of crowd control. USAID go out there, they're logistics people and goodness knows what else. And NIH have a few people who are getting some samples for some research. And we have the public health portfolio and do the things that we should do properly. And the one thing we can do and have started to do is training. And it's training of healthcare workers. The problem is there aren't enough healthcare workers to go around. And clearly the problems associated with recruitment of healthcare workers to go into West Africa at the moment really are great, especially with the kind of publicity that there is circulating around the United States at the moment. It's kind of bizarre. So just to get back to the subject of diagnostics, and I did have basically in the end two slides addressing the question of diagnostics. It's quite clear that, as Anita said, that the throughput of these PCR systems is just so slow as not to be of great value. Because, to be quite honest, if you've got a malaria endemic area and there are other infectious diseases which cause fevers as well, it's quite clear that you're going to be spending an awful lot of time doing tests, waiting for results, and how do you separate the people who might have malaria from the ones who have Ebola? And if you're putting them all together, if you didn't have Ebola when you went into the area, there's a good chance that you might get it before you get out. So there is absolutely a need for a simpler, more rapid approach to Ebola diagnosis. And antigen detection tests are obviously the things that matter. Unfortunately, unless there are either unique characteristics of the antibody which is used to capture the antigen, in other words, high affinity antibodies that can capture small amounts of antibody, plus signal amplification of the complex which occurs on those lines that you see on strip tests, then the chances are that you're going to lack sensitivity compared with the gold standard, which I don't know which gold standard one is going to use, but presumably it's going to be a PCR somewhere along the line. So what we really need, and it's echoing Anita's thoughts, is we need something that's going to actually be very sensitive, and as obviously these tests tend to be very specific anyway. That's inherent in their makeup, but very sensitive, but as sensitive as we possibly can, so that we can detect perhaps 1 day or 2 days after a real-time PCR becomes positive. I don't know we can expect too much more than that. And the important thing is that if there's somebody who has a fever, you can use this not only to diagnose Ebola, but to exclude Ebola. And people don't think about the use of rapid tests to exclude diseases. And I think that's something which needs to be remembered as well. So if you want details about the CDC's RT-PCR, about the way to transport specimens to labs, etc., and the standard operating procedures for RNA extraction and all the technical pieces, you can get it with this particular document. Now, I don't know whether we are sharing the slides, in which case you're going to get a copy of this anyway, so that's not so bad. Thank you very much indeed.
Obrigada, Dr. Ron. Agora poderemos dar seguimento com a palestra da doutora Sarvish.
Good morning, or should I say good day, Madam Chair, my esteemed colleagues, and ladies and gentlemen. Firstly, I'd like to thank Dr. Carlos Govadia for inviting us to come all across the Atlantic to this prestigious meeting, and I'm just privileged to share with you a snapshot of what we're doing in Africa. So we hail from Southern Africa, and my colleague is here, Patience Dabula. So just an overview of what's happening with Ebola and just a background on what the National Health Laboratory Service does, our current work, the National Institute of Communicable Diseases, which is a subdivision of our organization, what support we provide to the SADC region, the training interventions within our country, and the case study at Sierra Leone. Okay, so this is just a little map of where we are. We are right at the tip, so that's why South Africa. And it also breaks down our country into all the different regions that you see. And then I just wanted to show you quickly our footprint within the country. We have over 300 laboratory sites, and we have over 7,887 employees within our organization. And just an overview, NHLS is a public sector diagnostic pathology services, and we service 80% of the population, which is our 52 million people. And I did show you a footprint of our labs, which is over 300 laboratories in the country. And I wanted to bring to the attention, because we're speaking about IVD, what our operational budget is, and is about 3.5 billion. And the amount of money we spend on reagents and diagnostic equipment is equal to about $80,000 US. And of that, our technology spend is over $15 million US. So just to share with you, so you know what sort of buying power we have within the southern tip of Africa. And just an overview that there are 9 provinces within the national health, which is in South Africa, and of the 9 provinces, we have a designated hospital which is linked to a academic center that supports our Ebola outbreak. And these hospitals have district hospitals within their region that are also supported, and we provide them with training and support within the country. And this is PPE training, what the healthcare is supposed to do, and when you have a patient that comes in with fever, how we are screening. We're looking for malaria, we screen for the LFT, the FBC. These are just routine diagnostic screening tests that the healthcare worker would perform before sending the patient forward if needed. And our source of excellence is the National Institute for Communicable Diseases, where they have a specialized facility, which is a BSL-4 facility that these diagnostic tests are being conducted. So this is just a little picture of the BSL-4 facility. This is the only facility in Africa. So all the specimens come out to the NICD, and that's where we only perform the PCR test, by the way. That's where the tests are performed. And so what has happened is, with the outbreak of Ebola, the SADC region, which is all these countries that are neighboring South Africa, had a formal agreement where South Africa would support training, outbreak response, and just general support within this region. And that's where we've seen that this manual, I think we've seen a bit of a snapshot of where this manual is coming from that Dr. Ron has shown us. And this is the handbook that was distributed to help us, guide us with the Ebola and what to expect. And then we had PPE pamphlets to educate healthcare workers and anybody that would be coming into contact with patients that may suspect what sort of PPE to use. And it was just a simplistic flow chart, but was very informative. And then also basic PPE, again, how you put on the gown and what is expected and how you would prevent infections moving forward. So this was all provided with the CDC as well as WHO initiatives within our region. And I think you saw the handbook that we're going to be getting, and this was all came with that pack. So the NICD, as I indicated earlier, is responsible for surveillance, and we have a tremendous amount of support with WHO and CDC, and their BSL facility was the chosen facility to start the initial testing, and the testing I indicated were the PCR tests. So the NICD embarked on sending 4 staff members in a mobile unit to Sierra Leone to start the testing. So this is just, now I'm going to just show you quickly, and I know I have a limitation with my time, of our staff that went through and how we started our preparations and where we went from this. So this is all the boxes that they started packing for their trip to Sierra Leone, and also you can see, because they're all healthcare workers, the amount of drugs that they took for themselves when they've gone across there. You know, you always have to prevent on your own. And then this is us loading all these boxes into the containers that had come to fetch the stuff. This is all the equipment, and this is one of them. His name is Gunta. He is a specialist with biosafety, and he was busy loading the stuff and making sure that nothing is broken when you start loading. And these are the people, that's Dr. Pavaska and Chantal and Gunta, and these are the people that went, and these were their special UN clearance security tags that they were given at Sierra Leone when they as a support lab services staff. And then we went on to show, they just showed us a little bit of, already in Sierra Leone, what sort of preventative posters were out there, indicating to the staff what to do, what not to do, and the campaign of awareness of health in the country. And so when the guys got there, they started unpacking their boxes from this sort of container to this proposed lab area that they were allocated. And this is our team, you can see there. They starting to set up their mobile laboratory. And then we had, like all lab people, you have to modify things because it may have not fitted. So you can see Gunta is busy sorting out something there before it went into the lab. And this was our notice. I hope you can see it. It's just showing you that this is the mobile lab that was provided. And this is the team, proudly South African, and these are all the members that took on the task, and they specialized in hemorrhagic fevers. And this is when the president of Sierra Leone went to visit them. There was a big convoyage of people and a lot of activity. And this is now the lab. You can see the tent that has been set up. It was just an overview of what their facility. And you can see the pressure chamber. They called it the ISO arc, which is a negative pressure chamber. And then this is now when they started receiving all the specimens. It was received in packets like this, and then you had to start spraying them down and swabbing and at the specimen reception. And we started with Gunta's screening the actual containers, and these are the specimen reception, the specimen forms that the specimens came in with the patient details. And then this is actually the safety, the ISO tank, as we call it. This is our special safety facility where they started with the actual opening and preparing the specimen. And you can see Chantal is busy starting up the PCR and the test actually being carried out, and positive controls are important. And this is they're looking at the results, and we are just an overview of, trying to read the results and get the printouts. And yeah, so that's basically a quick snapshot of the support that we have, South Africa have provided. This is just to acknowledge the team that have gone through. And so in Africa, you always, as we say in Africa, it's called Ubuntu, we make a plan. So yeah, we South Africans, we made a plan, and this is our initiative to move forward in the continent. And I just wanted to say thank you. Okay, thank you.
Muito obrigada. Todos estão fazendo suas palestras em 10 minutos, significa que nós vamos ter bastante tempo para perguntas, o que é ótimo. Convido agora a doutora Vanessa para fazer a palestra.
Bom dia a todos. Primeiramente, agradeço o convite, ao Carlos, que nos convidou para mostrar a preparação do Brasil em relação ao surto de ebola que está ocorrendo na África Ocidental. E aqui eu trago um pouco dessa nossa experiência e também estarei disponível para responder alguma coisa que eu não tenha contemplado aqui na palestra. Então, na verdade, desde 2000, o Brasil ele tem adotado algumas medidas para fortalecer a capacidade de resposta e preparar-se para emergências em saúde pública de uma forma geral. E, principalmente com o advento dos eventos de massa, que aí entrou a Copa das Confederações, Jornada Mundial da Juventude, principalmente a Copa do Mundo FIFA 2014. Então, o Brasil veio se preparando desde o início para atender a eventuais emergências em saúde pública que pudessem ocorrer nessa época. E, nessa oportunidade, teve a questão de incorporação de novas tecnologias, ampliação de infraestrutura, sistematização de processos de trabalho na área de vigilância e resposta. Ali tem acho que apertando aqui. Esses são os nossos planos de contingência que estão disponíveis no site da Secretaria de Vigilância em Saúde. Existem planos de contingência para diversas doenças, para desastres, existe o nosso plano geral de emergências, a construção desses planos também foi uma preparação para as emergências em saúde pública. Aqui, falando um pouco mais da preparação do Brasil para manejo de eventual caso suspeito de ebola no país, mesmo considerando que o risco do Brasil de vir uma pessoa, um viajante dos países afetados para o Brasil, ele é muito baixo, esse risco, por conta que a gente não tem voos diretos dessas localidades para o Brasil, nem tem uma circulação de pessoas dessas nacionalidades aqui dentro do Brasil já comumente. Mesmo assim, o Brasil ele se preparou desde a declaração da OMS, de Emergência de Saúde Pública de Importância Internacional. O Brasil ele vem se preparando para lidar com a entrada de um eventual caso suspeito aqui dentro do país. E, para isso, uma série de medidas foram tomadas. Então, aqui eu cito um pouco da nossa preparação, que foi a elaboração de vários documentos técnicos. Ali eu cito alguns, ali o plano de contingência do ebola, que ele está em constante atualização. Na verdade, a gente tem um plano já elaborado, já faz um certo tempo, mas esse plano ele vem sendo continuamente atualizado com as novas recomendações, com os novos conhecimentos que vêm se adquirindo ao longo do tempo. Além disso, existe o protocolo de vigilância e manejo de casos suspeitos de doenças do vírus ebola, o plano de comunicação de riscos, procedimentos operacionais padrão para acionamento da equipe de resposta rápida, o guia de orientação para atendimento e remoção de pacientes, que é o atendimento pré-hospitalar, que a gente chama aqui o SAMU, que é o nosso serviço de atendimento médico de urgência. Existem orientações também para o manejo desses pacientes com suspeitos, no atendimento, definição de padrões para equipamentos de proteção individual, enfim, tem uma série de documentos, todos eles estão disponíveis no site da Secretaria de Vigilância em Saúde, que está reunindo documentos não só da Secretaria de Vigilância, mas também da Anvisa, que é o nosso órgão regulador. Além disso, foi instalado o Centro de Operações de Emergência em Saúde desde o primeiro dia da declaração da OMS de Emergência de Saúde Pública de Importância Internacional. Foi acionado esse nosso Centro de Operações de Emergência e ele continua em ativação e vai se manter ativado até que a OMS declare extinta a emergência. Inicialmente, a gente começou com 3 reuniões semanais, então, existem diversos participantes nesse nosso COIS, incluindo pessoas da Secretaria de Vigilância, pessoas da Assistência e da Anvisa, e também da nossa Secretaria Executiva do Ministério da Saúde. Essas pessoas estavam se reunindo 3 vezes por semana para lidar apenas com ebola. Isso tudo para que fossem discutidos os fluxos, os procedimentos, a definição do que ainda era necessário para lidar com o manejo desse caso, o que é que a gente precisaria resolver dentro do país para que o Brasil estivesse preparado para lidar com uma eventual entrada de um caso suspeito. E essas reuniões agora elas estão acontecendo 2 vezes por semana, porque muitos dos protocolos já foram estabelecidos, várias ações já foram realizadas e a gente diminuiu de 3 para 2 vezes por semana essas reuniões, mas elas continuam acontecendo. Também foi convocado o Grupo Executivo Interministerial para Emergências de Saúde Pública, que tem participação de todos os ministérios. Existem representantes de todos os ministérios que se reúnem 1 vez por semana para falar apenas de ebola e toda a preparação e tudo o que é necessário ainda ser feito para lidar com a entrada de um caso suspeito aqui. Também são realizadas videoconferências semanais com todos os nossos 27 estados. Normalmente é de quinta-feira pela manhã, todos os estados se conectam juntos e nessas videoconferências é um momento para a integração das ações que são realizadas, divulgação dos nossos protocolos, nossos materiais técnicos que são realizados, e eles também retiram muitas dúvidas. Afinal de contas, o ebola para a gente é uma coisa que está bem longe ainda de acontecer, né, aqui dentro do Brasil. Então, tem muitas dúvidas ainda que os profissionais têm, e essas videoconferências elas servem também para a retirada de dúvidas, estabelecimento de fluxos, protocolos com todos os estados. Também foram definidos, dentro do Brasil, nos 27 estados, hospitais de referência, né? Todos os estados definiram qual seria o seu hospital de referência numa eventual entrada de um caso suspeito de ebola no país, qual seria o hospital de referência que ficaria responsável pelo atendimento inicial do paciente. Além disso, foram definidos também hospitais de referência nacional, que esses hospitais de referência nacional são 2, que em São Paulo é o Emílio Ribas, e para o resto do país seria o Instituto Nacional de Infectologia, que é o INE, que fica no Rio de Janeiro. Na entrada de um caso suspeito, o que se orienta é que esse paciente ele seja encaminhado inicialmente para um hospital de referência estadual, se realmente ele não tiver condições de ser encaminhado diretamente para o Hospital de Referência Nacional. Mas é no Hospital de Referência Nacional que ele vai ter todo o apoio e toda a preparação realmente para seguir com o atendimento daquele paciente. Então, assim que possível, o paciente ele vai ser encaminhado para esse Hospital de Referência Nacional, que pode ser em São Paulo ou no Rio de Janeiro. Todos os hospitais de referência eles foram supridos com testes rápidos para malária e caixas de padrão NB3, de nível de biossegurança 3, para o transporte de amostras para o nosso laboratório de referência nacional. Temos apenas um laboratório de referência nacional, que é o Instituto Evandro Chagas, que fica no Pará, que ele é especialista nesse tipo de diagnóstico, e ele é que vai receber as amostras vindas de qualquer canto do país para a realização do diagnóstico para a ebola. Todos os nossos laboratórios centrais, nossos LACENs, que existem em cada estado, eles possuem profissionais certificados pela Organização Mundial de Saúde para o condicionamento de amostras de risco biológico de nível 4. Isso é uma potencialidade que nós temos. Então, independente do estado que aconteça o caso, nós temos profissionais habilitados que podem fazer a coleta desse material e o envio para o nosso laboratório de referência nacional, que é o IEC, no Pará. Além disso, foi ativada uma central telefônica gratuita, 24 horas por dia, 7 dias por semana, para o recebimento de notificações e orientações técnicas relacionadas ao ebola. Então, ali é o nosso 0800 644 6645, esse é o telefone que foi disponibilizado, que está funcionando 24 horas para recebimento de notificações. Além disso, nós, para testar todos os nossos protocolos que tinham sido elaborados e discutidos com os estados, nós organizamos exercícios de simulação, tanto no Rio de Janeiro quanto em São Paulo, foram simulados completos, chamados simulados completos, que envolveram desde a chegada de um suposto caso suspeito no aeroporto até o manejo desse paciente para o hospital de referência. Então, foram simulados bem interessantes, que foram identificadas falhas nos nossos protocolos, e a gente conseguiu reverter tudo isso, rediscutiu com os estados, muita coisa deu certo, e o que deu errado nós conseguimos alterar e modificar o nosso protocolo, e foi uma experiência bem interessante, não só para o Ministério da Saúde, como também para os estados e todo mundo que acompanhou. Isso envolveu a retirada de uma pessoa, da pessoa do avião, o transporte do nosso serviço de atendimento móvel de urgência até o hospital de referência. No hospital de referência, ele também foi adotado todo o procedimento para o manejo desse caso, e foi testado toda a cadeia, Além disso, nós tivemos também um simulado de mesa no Porto de Santos, que é considerando o Porto como uma possível entrada também de um caso suspeito. Nesse simulado de mesa, aconteceu com o Ministério da Saúde, a Secretaria de Portos da Presidência da República e a Companhia Docas do Estado de São Paulo. Então, foi um exemplo fictício do resgate de um suspeito a bordo de um navio que estava procedendo desses países afetados. Também foi enviado kits de equipamentos de proteção individual para os serviços de atendimento móvel de urgência, o nosso SAMU, das capitais. Na verdade, o envio, na época da Copa, de preparação para a Copa do Mundo, todas as cidades-sede passaram por treinamento em QBRNE, que é ameaças químicas, biológicas, radioativas e nucleares. E, na ocasião desse treinamento, também foram disponibilizados equipamentos de proteção para ameaças biológicas, que, no caso, o que a gente fez foi encaminhar para as outras cidades, que não foram cidades-sede, encaminhar esses outros kits de equipamentos de proteção individual. Então, aqui só algumas definições. O Brasil ele considera como caso suspeito, isso aí está disponível lá do nosso protocolo, indivíduo que veio procedente nos últimos 21 dias, que é o período máximo de incubação do ebola, de países com transmissão disseminada ou intensa do ebola, que aí, no caso, entra Libéria, Guiné e Serra Leoa, que apresentem febre ou esses outros sintomas, diarreia, vômito, sinais de hemorragia e esses outros sintomas. E também são considerados suspeitos os indivíduos que relatem contato com pessoa suspeita ou confirmada para ebola, que apresentarem sinais e sintomas citados. Então, essa é a nossa definição de caso suspeito, ela está disponível em todos os serviços de saúde, para que realmente se consiga detectar o quanto antes que aquele se constitui um caso suspeito e que todas as medidas do protocolo possam ser estabelecidas, assim como ocorreu no nosso único caso suspeito, que depois eu vou falar um pouco da experiência, como é que foi. Nós tivemos um único caso suspeito e que realmente o protocolo ele funcionou bem. E ali a definição de contactante ou comunicante, que é a mesma definição que a OMS trata, que são indivíduos que têm contato com sangue, fluido, secreção, isso aí a gente já sabe. Ali, em relação à notificação, que existe a notificação, ela deve ocorrer de forma imediata, segundo a nossa portaria de doenças de notificação compulsória. que é a portaria número 1271 de 2014. O ebola ele é uma doença de notificação imediata, ou seja, em até 24 horas tem que ser informado ao serviço de saúde a existência de um caso suspeito de ebola pela forma mais rápida possível, que, no caso, a gente orienta que seja pela nossa central telefônica, que é 24 horas por dia, 7 dias por semana. E também através do e-mail, notifica@saude.gov.br, e existe também um formulário específico que pode ser preenchido. Aqui é só mostrando um pouco o fluxograma do manejo do caso suspeito, que também consta no nosso protocolo. Então, um paciente suspeito de ebola, ele pode chegar no serviço de saúde por qualquer meio. Então, ele pode procurar voluntariamente um serviço de saúde, seja ele uma unidade de pronto atendimento, um serviço de urgência e emergência. Então, durante o tempo que durar a emergência de saúde pública de importância internacional, a gente está orientando que todos os serviços de saúde, frente a um caso que tenha febre e que seja procedente nos últimos 21 dias de viagens internacionais, se questione o local de onde essa pessoa veio. Então, identificando que essa pessoa veio ou da Guiné, ou da Libéria, ou de Serra Leoa, essa pessoa é identificada imediatamente como caso suspeito, e existem 3 ações imediatas a serem tomadas. Primeiramente, isolamento do paciente, mesmo que não seja um hospital que tenha um local específico para isolamento, esse paciente ele precisa ficar isolado, nem que seja numa sala, num local que ele fique totalmente isolado de outras pessoas. Além disso, tem que ser feita a notificação imediata do caso para a Secretaria de Vigilância, para a Secretaria Estadual de Saúde e para a Secretaria Municipal também. E também contactar o nosso SAMU 192, que é o nosso serviço que vai fazer o transporte desse paciente, de qualquer que seja o local onde ele esteja, qualquer que seja o serviço de saúde, o SAMU vai lá buscar a pessoa que já vai estar em isolamento e levar ela para o hospital de referência estadual. Isso, se não for possível, levar diretamente para o hospital de referência nacional. Então, o SAMU, o que ficou sob responsabilidade do SAMU é que ele vai receber a ligação, né, informando que existe um caso suspeito, seja ele dentro de um serviço de saúde, seja dentro do aeroporto, por exemplo. O SAMU ele vai pegar essa pessoa e vai levá-la para o hospital de referência estadual. Aqui é um fluxograma em relação ao aeroporto, né, que, a partir do momento que existe a informação da presença de um caso suspeito dentro da aeronave, a gente sabe que a OMS está orientando que, na saída dos casos, na saída das pessoas desses locais de origem, de Guiné, Libéria e Serra Leoa, exista uma triagem para que não seja possível viajar pessoas que estejam com algum sintoma ou que tenham tido contato com casos confirmados ou suspeitos de ebola. Então, essa é a triagem mais efetiva. No entanto, no momento da vinda dessa pessoa, ou até dentro da aeronave, ou assim que ela chegar, que ela desembarcar, ela pode iniciar os sintomas. Então, nesse caso, tem que haver um contato diretamente com o nosso posto da Anvisa, que é a nossa vigilância sanitária. Dentro de todos os aeroportos existe o posto da Anvisa, e esse posto da Anvisa vai fazer a comunicação, ligar para o SAMU, para que ele esteja de prontidão no momento que o avião chegar em solo. O SAMU ele vai estar de prontidão para levar essa pessoa para o hospital de referência. E, além disso, a Anvisa ficou responsável também por fazer perguntas. No caso, existe um questionário para ser perguntado a todas as pessoas que estavam nesse voo. E as pessoas das fileiras, uma fileira anterior, uma fileira posterior e as fileiras do lado, são considerados contactantes, e as demais do voo, elas também são questionadas, porque pode ter acontecido alguma coisa no voo que elas se constituam como também comunicantes. Aqui é o nosso fluxograma do Hospital de Referência Estadual. Digamos que esse hospital recebe um caso suspeito, ele vai fazer novamente a notificação imediata, mesmo que essa notificação já tenha ocorrido no serviço inicial que o paciente recebeu. recebeu o primeiro atendimento, que foi identificado como caso suspeito. Mesmo assim, por redundância, se pede que esse hospital de referência estadual faça a notificação imediatamente, assim que ele receba um caso suspeito. Além disso, ele vai fazer orientações ao paciente e aos seus acompanhantes, se for o caso, e isolar imediatamente esse paciente. No momento do isolamento, vai ser verificada a necessidade de hidratação oral desse paciente e tudo mais, outras coisas. Realizada antibiótico terapia com ceftriaxona. E esse paciente ele vai ser, a todo momento, reavaliado para ver se existe possibilidade de transferência desse paciente para o Hospital de Referência Nacional. Então, a primeira coisa que chegou o paciente, após o isolamento, vai ser realizada uma análise desse paciente, uma análise médica, para saber se ele tem condições ou não de ser transferido para o Hospital de Referência Nacional. E, assim que ele tiver condições, ele vai ser encaminhado para o Hospital de Referência Nacional. Caso ele não tenha condições, ele vai seguir na cadeia com a coleta de amostras. Vai ser coletada a amostra para o envio para o nosso laboratório de referência nacional para a realização do teste diagnóstico, o RT-PCR para o ebola. Além disso, é realizado um teste rápido para a malária, que todos os hospitais de referência eles receberam esse teste rápido para a malária. No entanto, mesmo que o teste rápido para a malária identifique que ele está com malária, não vai ser descartada a possibilidade de ter ebola também. E aí é enviada essa amostra para o nosso laboratório de referência estadual. Essa amostra dando negativa, é esperado um tempo de 48 horas da primeira coleta para a realização de nova coleta, e, dando novamente negativo, esse caso ele é descartado e é desmontado todo o esquema. Se na primeira amostra já der positivo, na primeira ou na segunda amostra der positivo, ele é um caso confirmado para ebola. Aqui o nosso fluxograma para o Hospital de Referência Nacional, que é exatamente o mesmo, só que ele já segue diretamente com toda a assistência ao paciente, ele não tem aquela avaliação contínua para saber se precisa enviar para o Hospital de Referência Nacional, porque ele é o Hospital de Referência Nacional. É exatamente no Hospital de Referência Nacional que vão ser realizados todos os demais exames diagnósticos que precisem ser feitos, né, e exame patológico, enfim, qualquer que seja o exame vai ser realizado no Hospital de Referência Nacional, e segue todo o fluxo que eu já falei, igual ao Hospital de Referência Estadual. Aqui é só a descrição do nosso único caso suspeito no Brasil, que ele ocorreu agora há pouco, né, agora no dia 9 de outubro. E aqui tem a descrição exatamente como foi a saída dele da Guiné. Exatamente no 21º dia, foi o dia que ele foi para a nossa unidade de pronto atendimento, lá em Cascavel, no Paraná. Então, lá em Cascavel, foi identificado, foi questionado se ele veio de viagem internacional dos últimos 21 dias, ele disse que sim, que veio da Guiné. Então, a partir desse momento, a UPA de Cascavel, eles fizeram até mais, eles não isolaram o paciente, eles isolaram a UPA inteira, né? Eles fecharam totalmente o serviço, tiraram as pessoas que estavam em outra ala, mandaram para outras localidades. E quem estava no mesmo local que o paciente foi atendido, e todo mundo que teve, de certa forma, algum contato com ele, ou passou pelo mesmo local dele, foi isolado também, para que pudesse passar por questionamentos, né, da equipe de investigação. E esse caso, num dia depois, menos de 24 horas, ele já estava no nosso Hospital de Referência Nacional, no Rio de Janeiro. Lá eles seguiram todo o protocolo, tudo exatamente como tinha sido previsto. Foi realizada a primeira coleta, enviada para o IEC no Pará. Essa primeira análise deu negativa. Depois de 48 horas, foi realizada nova coleta, a segunda coleta deu negativa também, e, a partir de então, o caso foi descartado. No entanto, uma equipe de investigação, nesse momento aqui, logo quando ele foi identificado, ele foi no dia 9 de outubro, ele foi para a UPA, no entanto, ele relatou sintomas desde o dia anterior. Então, todo mundo que teve contato com ele no dia anterior e no dia 9, que foi o dia que ele foi para a UPA, todo mundo foi questionado, foi investigado, foi realizado a equipe de resposta rápida do Ministério da Saúde, junto com a Secretaria Estadual e Municipal, fizeram questionamentos a essas pessoas. No total, foram 201 pessoas que foram identificadas, sendo 131 os indivíduos que passaram pela UPA naquele dia, sala de espera e pessoas internadas, 19 que dormiram no mesmo albergue que a pessoa estava, que o rapaz ele estava hospedado, e 51 profissionais, entre a UPA e também o albergue. E identificaram 3 que teriam maior risco de exposição. E todas essas pessoas foram monitoradas e não tiveram febre até o descarte do caso. Então, quando o caso foi descartado, toda a mobilização que estava ocorrendo para aqueles casos contactantes, ela foi desativada, porque, na verdade, o caso não foi confirmado, né? Ele foi descartado. Então, só para finalizar, essa é a maior epidemia de ebola da história, como a gente já sabe, né? A doença ela nunca atingiu grandes cidades, e era característica muito mais de populações isoladas. Essa disseminação, existe risco de disseminação para outros países, no entanto, para o Brasil, a gente considera que esse risco ele é extremamente baixo, né? Mesmo assim, todas as medidas que o Brasil está adotando para evitar que, na eventual entrada de um caso suspeito de ebola aqui, que não haja nenhuma outra pessoa, nenhum segundo caso decorrente dessa pessoa que entre. Não tem como a gente impedir que chegue uma pessoa infectada aqui dentro do país. Não tem como a gente impedir isso, até mesmo porque não existe nenhum tipo de proibição ao comércio, nem a viagens para essas localidades. Então, não tem como a gente impedir. O que a gente não quer é que não tenha caso nenhum decorrente desse primeiro caso. Esse é o nosso objetivo, né? E aí, com esse conhecimento que a gente adquiriu, a gente pode prevenir e, com isso, a gente pode ter saúde. Tá bom? Obrigada.
Obrigada, Vanessa. Com a palavra, já vamos.
So I assume I'm from industry and I want to try explain what our solution is. It's good to come the last. So I want to make a couple of quick remarks on it's linking to the discussions which Anita from WHO and Ron from CDC mentioned. And I think is coming from a diagnostic company, we don't see Ebola anything different than others. The panic, the discussions going out there, we understand that's happening now, but for us, Ebola is like another diseases but we have been in our portfolio we're working. Obviously, the level of the publicity has this got is enormous. I don't think we have been so much in the spotlight since we have been involved with Ebola. So this is good or bad. The bad is that just impacting our other program we have. But the good is that we were working with febrile illness. So that is the one aspect on that. The other thing is, what I will mention is that we have to see this as a long-term commitment, which I think sometimes doesn't exist. And putting a diagnostic test to the field is a challenge. While you can make a diagnostic very quickly, right or wrong, to keep it years after years on the quality you want, it takes commitment, and we do know that. And that's the reason I want to start. If you can just bring the light a little down, please. As I mentioned yesterday, like Ron mentioned, we have a commitment to help people live longer life and healthier life. So as this happening in US, as a US company, we want to try to contribute as much as we can, not only for this epidemic happening right now, but the next epidemic happening, which is just going to happen. With these global changes, climate changes, we do see every day. I have been so many years talking about the Chagas for US. Oh, we don't have a Chagas in US. Chagas never come to US. But that's wrong. We have studies done in southern states in US. We have tested dogs. We have tested the immigrants. We have tested the population. We can really show Chagas exist. We have been in Mexico. I was 3 times in Mexico this year. We are trying to convince Mexican government to go with the Chagas screening. They don't want to do that because they think the Chagas doesn't exist. So these diseases, chikungunya, dengue outbreak, all of them coming one after each others. So now Ebola is a little extreme, and that's the reason you get the publicity. You get bleeding, external, internal. You have to shut down the airport. You have to get everything. So that's a different story. But in general, the infectious diseases, these global diseases, is just a question of time. They are going to happen and happen and happen. So being a part of that, I think as our company, we have a very good global networking. That's another part of the business, but that's part of a strategy. We have been working with a group in Peru, Segunda is one of them, but we are doing actually a febrile illness testing program in tropical area in Peru last 6 months in Navy lab. We are doing some study, as I mentioned yesterday, in Sierra Leone. It's a very difficult to go. We're doing in Thailand. We do in other places. So these global networking, it's extremely important to be in the right and with the right people. In my experience, the last 25 years, if you are not partnering with the right people, the right team, you are for short run. And that's another good start. I don't want to talk about the HIV, but I put it here just highlighting again, if we come with a solution, we think we can make this product. And that's what highlight, we have to live up with the standard of the global regulatory body, which is, I think, is the WHO is one, but there are many of them. It's a CE, FDA, and other things. So I think we are confident whenever we have the solutions, while we live this panic situation out, I think we can have the right solution to be it. I will talk about the Brazil as well. I think it was good to come after the Brazilian presentation. We have been in Brazil 10 years. We are one of the company with our partner Fiocruz Biomanguinhos. We innovate. We have introduced a test for the screening of dog for leishmaniosis. They are producing 2 million devices, testing pretty much every dog here, eradicate the leishmaniosis. So I don't see that disease is different than in other diseases. So while we have Ebola here in Brazil or not, but we have many other tropical diseases. Yeah, why I put this slide here? Because I want to go back to this. Because there is a link here to Ebola, not because of we have oral fluid HIV tests, we do it in Brazil through our partner in America, but the collection device we have a patent on that, and I think this is very valuable because we use this in our current effort to include in Ebola, particularly for scientifically a couple of reasons. This is a closed system, we want to treat the virus. There are some ideas we are working on that just right now to inactivate the virus, inactivate the sample, actually making the testing much more user-friendly and less risky. Break down the viruses, it is important. We want to go to blood. The blood matrix is different than the plasma, which PCR is using. The other is that we have a collection device. You can actually collect the sample, not only of the blood, but also with other body fluid. We saw the South African teams, they showed how the lab they're setting up. I have been in Africa, in Nigeria, Tanzania, and I have worked in HIV labs there. I know what complexity is. Running in the lab in Nigeria was a nightmare. That was actually in capital city. I was not in the remote area. So you can collect the sample of body fluid. It could be vomiting, it would be urine, it could be other thing. Then you close the system and then you can send the lab to run the assay. It could be PCR, it could be rapid test. So our goal is not to complement or replace. So let me just be careful about, clear that, and we don't need to go back. We see the diagnostic complementary. ELISA, PCR, rapid test, all that's part of the algorithm. It's not correct for me to, oh, you know what, we want to place a test, it will be as good as this PCR. No, it's not going to be that. But as a part of the solution, we have to do it, as I think the previous speaker talked about that. There is a need for rapid tests, as the same lab that we show in South African team put it in Sierra Leone. Because we were doing in 500 people in Sierra Leone, a similar setting. So now samples come, patients come, you can have other assay can help you to that. While you have this sophisticated, very sensitive, I think we can be right behind from a performance standpoint, but it's an overall value, it's clearly. Obviously, Ebola is nothing new. It has been from 76. It started with the Congo, and it is now spreading. It is a lot of other Ebola outbreak has happened. This is a slide. We are working, and I will tell you why we went in this area with the groups. And as you can see, 38 years has been this. It is basically year by year what's happening. The total and the death. That you can see the dynamic happening now 2004. And these are the model, now the CDC and other group working to see how they can have under control. And then you can see here's the reproduction numbers. Ebola is not only the Zaire one, it's a 5 different type of that. And again, when next epidemic happening, someone need to differentiate the things. And I think that with our technology we have, we can actually contribute here. The Zaire one, the Sudan one, the Marburg one, the Uganda one, they changed the name. The government pressed them to change the name to Bugun. So that's all happening again and again. So I stop here and go back to what I think Ron mentioned, something very important. And I think this is I have seen, and some of our collaborators here from other institute in Brazil is the diagnostic tests live and die with the quality of the reagent. That it is. I don't know how many calls I get every day or every week on, look, we need this test, we need that test, we need the other one. The first question we always ask, do you have the reagent? Do you have the well-characterized reagent that can pick up what you want and discriminate what you don't want? If you have that, then we can make a diagnostic test, no problem, okay? But if you don't have that, it's a short shot. You will just make a test, which you can make it. There's many assays, you make it, you can produce a one lot, the group's making a good reagent. Oh, I have fantastic reagent in my laboratory, and I can make, 5 milligram of that. When you ask them the next batch, they cannot produce the reagent. So when Ebola happened, while it was a part of the program we were working, we had a networking to go to right people. The people working with Ebola vaccine, Ebola drugs, identify reagents that can target the virus. Because why? Because currently the drugs has been used is based on the monoclonal antibody. They're targeting these things, okay? And if you have a right reagent, then it's a good start. So that was being, unfortunately, I have missed the deadline for Anita for our application, but we have been working very carefully on the radar to really link us with a partner to identify a reagent can be used, that we don't promise something that we cannot back it up. So that's the reason we enter a week or 2 ago. We have been working with the groups that they have the reagent that has been used in a drug. And that was the reason we identify things. So it come to our mind, basically there are potential with few product here. So one of our classical point of care is that we think is an antigen detection system. You could go after blood, serum, plasma, urine, oral fluid, contaminated material, all of them. And all these matrices is actually very, very important because you want to control the spreading of the disease. And we do see how this is spreading. I can give a greatest example. Animal testing, it's become very critical. Last night when I'm sitting in my hotel, go to dinner, I get a call from US, one of the largest veterinary company in US, and asking, helping them in Ebola for animals, which is issues. We did see publicity in Spain that their dog was infected. Can you believe it? It's a lot of hysteria going on. There are other assays after epidemic goes, you can actually monitor serological aspect. It's very important. We have been doing this type of assay for dengue, other diseases. Why you need that? Because in this endemic area, you can actually have 10 to 15% of the people have the Ebola antibodies without being infectious. So from a monitoring aspect, it's going to be important. Maybe there is a value to monitor the drug's effectiveness. So IgG, IgM come behind antigens, like in dengue disease. You detect the NS1, you detect IgM, you detect the IgG. Then by reviewing those 3 markers, you make an assessment. Is the new infection, is the co-infection, it's a reinfection, so all kind of thing. And then is obviously a third format that I think that's what actually we started, and that's what I think it will stay forever, especially in Brazil and many places. This is what actually Brazil will need. That's what I talked to our partner. We think that if Ebola exists or not in Brazil is irrelevant. But there is other diseases exist. Chikungunya, dengue, yellow fever, malaria. All of them exist in Brazil. Now, we can have another ban for Ebola there, you see it or not, doesn't really matter. 90% or 95% of diagnostics is always negative. They can detect something, but they never exist. So being a part of the febrile illness, it is actually the solution. So I will share some, because I know the certain group here is a scientific and technical group. I thought that basically this data generate as I speak, and I get update of my team pretty much every day what they're doing. So when we identify, we had a mass screening, probably we contacted everyone has the reagent around the globe. Hundreds of the reagent arrived, and we screened them. We have a system to screen them to identify which one is good and which one bad it is, with a reader, with a specificity. So this is a illustrative one of the data we have about the best pair. What I show here is we don't have today, but we are actually working very hard to get this BSL-4 labs and the wild type of Ebola. Hopefully within a week or two we have that. But these are a non-infectious material with a 3 level of we have found. We identify a group had a killed virus, Zaire, we got that. We had a recombinant and artificially virus particles. So here is affecting of the volume, because when you go to antigen detection, you have to increase the volume. As much as volume you can add there, you have more virus. So this graph is illustrated here that if you look at there, the graph green, it's showing the volume if we increase it to 150. So most of the point of care assays using 10 to the 50 microliter, we can actually increase to the 100, 150 microliter. That's one of the advantages with our platform with having a separate delivery. So we think that you can see the graph that right now we are heading around actual virus particle around the 0.5 nanogram, but we can go lower as well, okay? This is another material, virus-like particles we used again. You can see the effect of the volume. You can see the graph, how the difference is. The blue graph is 25, the green one is 150. So we think that to get the most sensitive and probably right behind the PCR, we need to go for larger volume. This is a killed virus. This is basically, it's showing that we can grab the epitope even the killed virus. This is actually the differences we did see in combination with this graph that we're looking for other. I have even seen some other assays right down, they are cross-reacting with the other Ebola virus. But if you look at these 4 graphs, we have more graphs to show, but I'm not showing here. It shows the value of having the right reagents, because we can see already we can detect recombinant, killed virus, virus-like particle. So we have targeting right area of a virus. And when we screen all these antibodies, we did see differences here. We did see some antibody was grabbing the virus, but not antigen virus-like, some recombinant. So we tried with the groups we are working, which they have been access to the key reagent through Army groups. They have been working with Ebola over 10 years. So that was an excellent connection we had through them to understand what's going on here. We are doing some modeling now as I speak. So this is just, we are trying to identify a model. I think this will be probably helpful for WHO group as well. They are trying to, how many, again, we are trying to look at how many particles, epitope, infection stage, where we are. So that our best assessment right now working actually with the leader in Ebola group is that the PCR is detecting around 2 to 3, 10 to the 3 PFU and PML. We think right now with our calculations, with the volume we use, we are at 10 to the 4, like 1 log behind the PCR. But we have not done actually optimization. As you can see, I highlighted some of this material there. And I think what we will know, that when we take devices to do BSL-4 labs, we will experiment to see what is the limit of detection is. And those are the studies, very difficult to do it, but we have to do them. I showed yesterday, again, just a few slides from the repeat yesterday, but I want to try to finish up with this. Again, the febrile illness is the way to go. So the 5-plex assays we had, you can see the list of there that the malaria was included there, HRP2 for early marker, PLDH, NS1, dengue, plague, Burkholderia. Those are the 5 markers we had selected. And I show you some slide in the end why we picked them. Now, one of the fast track we did, we add the Ebola #1 as well. So this is what we are trying through our connection get to Sierra Leone right now. So similar, the lab that we had, we showed in the South African teams that we were doing 6 months of clinical trial in Sierra Leone, not with Ebola, but is right in the same clinic which hit by Ebola. So they gave us the data. I actually received the data a few weeks ago. We are reviewing that. Now, you can see here that, like, look at again the Brazil. Malaria exists here. So you need to have the malaria in there. I will not go through that because I talked yesterday. Other febrile illness. So NS1, there's a lot of NS1 assays here, but not all of them is good. Some detect 1, some 4, some is strong with a 2. So we work with this funded program for Army to develop a right market. We end up putting 8 different monoclonal antibodies together to get the best sensitivity because they want to detect early as possible. We could do actually with the 2 antibodies, but it was not sensitive enough. Again, the reagent, you have to partner. We have partnering with Pasteur Institute, with other institutes, to really access to the reagents you can get for the plague. No one has the right one, so you have to put them together. Biothreat assessment is a part of this global. Obviously, some of diseases could be, like anthrax, would be non-biothreat as well, some regions, but you need to put them together. And that's the reason these multiplexings come in the pictures. Controlling the data is important. I showed yesterday this data for you guys, but in the end of the day, this is my last slide, which I started yesterday, we have to look at for these type of assay. I listed here. I want to just show this before I finish it. These are the list of the assessment we will make for different pathogen. You can see that you can put them together. It might be the region differences that in Brazil you have to do differently than Asia, but at the end of the day, that's the same stories. Okay? That's all.
Obrigada, Javan. Temos pouco de tempo para algumas perguntas.
Parabéns a todos, acho que foi extremamente interessante e importante essa discussão, especificamente sobre ebola, mas ela traz uma luz também para outros tipos de diagnóstico que podem surgir de uma forma emergencial ou outros diagnósticos que são importantes também numa entrada. Aqui no Brasil, nós temos uma situação específica da legislação. Então, a OMS, no caso a Anitta mostrou bem, está fazendo uma força-tarefa para avaliação em caráter extremamente urgente dos testes disponíveis e, num período de 3 meses, deve chegar a uma conclusão do que poderia ser pré-qualificado como interessante para se usar em vários países. Aqui no Brasil, a gente tem uma situação que uma resolução da Anvisa permite, a resolução 03 de 2010, ela permite que, por exemplo, numa situação de interesse público, o Ministério da Saúde pode solicitar um fast track para avaliação de determinado produto. Eu gostaria, se, na verdade, isso acho que é alguma coisa interessante, se a doutora Vanessa pudesse comentar, e também, Marcela, se você pudesse comentar, no caso específico, se fosse solicitado pelo Ministério, como vocês imaginam lidar com essa situação, imaginando também um estudo de caso.
Bom, em relação a especificamente essa área, não é a área de atuação assim minha especificamente, é mais a área de laboratórios. No entanto, o Ministério ele tem todo o interesse, na verdade, na identificação de testes que possam ser utilizados o quanto antes possível no diagnóstico, né? Isso aí é uma coisa que não tem dúvidas em relação a isso, né? É só essa questão da pré-qualificação que é essencial também, porque a gente precisa entender, exatamente saber quais são os testes que a gente pode utilizar, né? Quais são aqueles testes que têm uma sensibilidade, uma especificidade boas para que a gente possa utilizar com tranquilidade aqui dentro do Brasil. Talvez ela possa explicar melhor.
Então, alô? Oi. No que diz respeito ao registro dos produtos, é como o Carlos colocou. Ele pode ser analisado com prioridade, né, mediante a manifestação do Ministério da Saúde, mas aí a demanda parte do representante, enfim, do importador ou do fabricante desse produto. Então, a área teria que avaliar quais foram os estudos que foram apresentados, que foram realizados, enfim, avaliar todo o dossiê no trâmite que já é previsto na legislação para registro de qualquer tipo de produto, para poder conceder ou não o registro, né? Enfim, mediante uma solicitação. A análise, a manifestação do Ministério da Saúde, ela permite a avaliação em caráter prioritário. Então, a ordem normal é a ordem cronológica para análise, então, ele foge exclusivamente da fila, né, de análise, ele é analisado com prioridade, mas tendo que atender a todos os requisitos para a obtenção de um registro. Uma outra alternativa também que existe é a possibilidade de entrada de um produto sem necessariamente estar registrado na Anvisa, né, que é quando ele já tem uma avaliação por um organismo, ele é adquirido pelo Ministério da Saúde para programas de saúde pública, né, sendo adquiridos por organismos internacionais. E aí eu acredito que a pré-qualificação da OMS tem um papel importante nisso. Então, em medidas emergenciais, o Ministério da Saúde tem essa prerrogativa também de adquirir um produto, não necessariamente sendo avaliado na Anvisa, né, mas ele já vai ter uma qualificação reconhecida.
Mais alguma pergunta?
It's just a question for discussion, and that is we've spoken about tests for diagnosis, but of course not everybody with Ebola dies, and so consequently you need a test also to monitor when they're safe to go back into communities again. And at the moment, we use PCR in, let's say, the United States by doing 3 PCRs. And if they have 3 PCRs which are negative, then everybody says, thank you very much, and you can now join your family and be seen on TV being hugged by all your doctors. I think it's very important that if we have antigen tests coming out, that you're not going to be able to use PCRs on all the hundreds and hundreds of people who have been infected. We're going to have to look very carefully at what criteria we use these tests, if they ever come to the market, how we use them in order to release people back into the community as being non-infectious, because undoubtedly they're going to be less sensitive than the PCRs. So I think we must not just look in terms of diagnosis, but also other usages for these tests as well.
Mais alguma pergunta? Alguém da mesa gostaria de se manifestar? Não? Então, agradeço a todos. Convidamos para o almoço, então. Voltamos daqui exatamente a 1 hora. Obrigada.